Okay, today's lecture is going to focus on the brain and cranial nerves. I love the brain personally. I think the brain is absolutely a fascinating organ, and the crazy stuff that goes on with the brain is just fascinating. So I hope you enjoy it as much as I do. Hopefully, I'm going to try to throw in some interesting stuff here as we go through the actual lecture. So let's dig right into it, shall we? So the adult brain weighs approximately 1.4 kilograms. So for those of you that don't actually function on the metric system, that's approximately 3 pounds. It's got a volume of about 1,200 cc's, but cc stands for cubic centimeters. Um, by the way, um, something important to make note of that the, the size itself is there really only for your general knowledge. In terms of relative importance of the size of the brain, it matters very little because it's not the physical size of the brain that determines your intelligence or an organism's intelligence or a person's intelligence, but rather the number of active synapses that exist in that brain, right? So again, don't get hung up on the actual size of the organ per se. And by the way, we're talking about an adult brain here, not a child brain. Um, One of the most interesting aspects of the brain is the fact that it responds to stimuli with such great versatility and adaptability. It's very impressive in the way that it's very versatile. So it can respond to stimuli, the same stimulus in different ways. It can learn new stimuli, an incredible amount of different stimuli, and how to respond to those different stimuli differently in different situations, all depending on the person and the situation and environment they're in. One neuron, this one blows me away, by the way. One neuron can process information from about 100,000 different sources at the same time, which again is insane. I'm not entirely sure how that is possible, but yet it can do it. So it's very impressive that way. We've got a lot of neurons in the brain, about 20 billion, right? Give or take a million, who knows? But a lot of neurons in the brain, right? Because that's what the brain is. It's a centralized integration and processing center. Now, the brain, while it has this incredible versatility and this great, incredible adaptability and so many connections via neurons, that its functions are so amazing, but yet there are limitations to this organ, despite the fact that it is, has all these abilities. The brain cannot be both immediate and precise and adaptable all at the same time, right? So it cannot be all three of those at the exact same time. It can be immediate and precise, and the analogy I like to use here is like learning a new job, right? Many of us work, right? And so when you first get a job, nothing you do is going to be immediate because you're just learning that job for the most part. Most of us, when we get a new job, it's a new experience for us, very new experience for us. Very rarely does somebody get a new job doing the exact same work they were doing and they really have nothing to learn. So nothing you're going to be doing is immediate. Your work can still be precise. So that is, your responses by the brain can still be precise or precise. They can still be precise, and they can be adaptable, but they cannot be immediate as well because that adaptability doesn't allow for immediacy, right? But once you've been at that job for a while, you probably know that job in and out. You know exactly what you do, you know exactly what you're doing every step of the way, and so the immediacy is there. You can do that job precisely and immediately. But the second that something changes from, say, like a rote situation or a typical situation, and adaptability must play in now, all of a sudden that immediacy goes right out the window because you can't, again, do things immediately and adaptably at the same way. It's also difficult sometimes, depending on what it is you're doing, to be both precise and adaptable at the same time, right? Because if you don't know how to respond, then precision often becomes difficult. So lots of things to consider there. Still very interesting, though. Still very amazing what the brain can do despite its limitations. Uh, This figure in and of itself might be one of the creepier figures in your book. I especially do like the fact that there are eyes here for whatever reason, but that's neither here nor there. This picture will be more for reference. We'll come back to it later um, on as we go through certain important things. Okay, so the gray matter, right? When we talk about gray matter, and again, if you think back to the nervous system lecture that we've talked about, where the, 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 the actual neurons themselves, unmyelinated neurons, right? So this again specifically neurons with no myelin sheaths around them, they appear gray specifically because of the fact that they have these Nissl bodies in the cell themselves. Nissl bodies, remember again, are the ribosomes within those cells. And so your average neuron that's not myelinated is going to look gray. And so any gray matter within the brain, and typically, again, we're talking about the external sheets of the cortex, or the cerebral cortex. We're talking about all these external sheets here, where you see all these folds on top of the brain, right, that cover most of the surface of the adult brain, as well. And this, by the way, is the cerebrum. So that surface of the cerebrum, as well as the external surface of the cerebellum, which is right here. So the external sheets of these two portions of the brain are gray matter, right? And it's because gray matter houses motor neuron and interneuron cell bodies. So these are areas where information is going to get processed, right? It houses dendrites, which means information is coming in here. It's got axon terminals, right? So information is going to leave here as well. And then unmyelinated axons. So we're looking at all these unmyelinated aspects of a neuron, which is why gray matter is gray in the first place. Right? So again, when you think of gray matter, you think no myelin whatsoever. Well, that leaves us with white matter. And white matter is actually very easy now that we've had this conversation, because white matter is entirely myelinated axons. So in this case, I mean, you think back to what we know about an axon. The function of an axon is to carry a nervous impulse from the cell body all the way to the terminal end of that axon. That is to another neuron, or to an effector of some kind, whether that effector be a muscle or whether it be a gland. Right? And so we're talking about myelinated axons here, which means all white matter is basically a transportation highway, and that's it. It's just conveying information from one point to the other. So within within white matter, you should not have any actual processing that occurs, right? And in this case, when we look at the adult human brain, we see that the white matter is actually deep to the gray matter. So it's deep to the cortex. Cerebral cortex is here. This is ultimately what we often associate with the higher level functions of the brain. We're going to come back to that, talk about that. Now, occasionally within the masses of brain, or within the masses of white matter, you will see these clusters of gray matter, and we see them here and here, here and here, here, and again, obviously, symmetrically on both sides. And these clusters are called cerebral nuclei, right? These are clusters of gray matter. And from here on out, you want to make a note. From here on out, a nuclei or nucleus no longer means an individual nucleus within an individual neuron. Now, when we talk about nuclei or a nucleus, we are talking about a grouping of neuron cell bodies in one specific place. So this area right here is a nucleus, right? And this is a nucleus, if that makes sense, right? And so these are specific areas that we see are aggregated neuron cell bodies. So they're basically islands where information processing can occur within the brain. Now, before we actually talk about the brain itself to a great extent, and the function of the brain and all the interesting aspects of the brain, we do have to talk a bit about the support and protection of the brain that exists around the brain. So obviously we've got the skull superficially into the brain, right? So we've got the skull, that hard, bony cranium that provides that rigid support, that rigid protection around the outside of the brain. But once we get through the skull, this is where things become unique and different, and what we haven't talked about yet. And so these tissues that we call meninges start to become extremely important. A meninges is a protective connective tissue membrane that is there to both surround and cover, protect, and partition portions of the brain. So it's going to actually surround the entire brain. And it's going to divide up the brain into specific portions. We're going to show you where that happens. And within the spaces that are formed by these layers of meninges, we see cerebrospinal fluid. Cerebrospinal fluid is abbreviated CSF. So every time you see CSF now, you can think back to cerebrospinal fluid. This is a cushioning fluid. The brain itself actually floats within the skull. And so this provides a cushion, a barrier, so that the brain doesn't slam into the front of your head every time you run into a door, for example. Right? So that's another layer of protection that we have, but it's besides the meninges. And then finally, we also have that blood-brain barrier that we've talked about, that BBB that I referred to back again early on in the nervous system lecture. And the blood-brain barrier is, again, meant to prevent entry of these harmful things that we ingest. So whether it be harmful levels of toxins that we ingest, that toxin could be a drug, it could be a prescription drug, it could be a non-prescription drug, it could be an actual drug, a legal drug per se. It could be just changes in fundamental ions or hormones or other things that could damage the brain if we allowed them to come into the neurons. So this allows us to actually separate that out, make sure that those things don't affect the function of the brain. Okay, so when we talk about the meninges, we have to start by understanding the actual order of these meninges. So we're going to start superficial and run deep. So keep that in mind. The superficial most layer is the dura mater. Now, the word itself actually matters. Dura in Latin means tough, and mater in Latin means mother. So this term literally means tough mother. The dura mater is a very strong, fibrous, very cloudy white layer of tissue. It's very, very tough to cut. And so it literally holds up to the name tough mother. Now, that is the superficial most layer of meninges. Just deep to that is a space between the dura mater and the next layer that we're going to talk about here in a second. And that space here is now called the subdural space, which again, that seems to make sense. That space is beneath the dura, subdural. Okay, so we've got the dura mater, and then we've got the subdural space. And our next meninge is called the arachnoid meninge. And for those of you who are aware of this term here, arachnoid refers to spider. Now this doesn't look like a spider. It's called the arachnoid mater. It doesn't look like a spider, but it looks like webbing. And hence, because it looks like webs, it was named after that appearance. And so that's why we see the term arachnoid referred to here. This is called the arachnoid or the arachnoid mater. Again, deep to the arachnoid mater is a space. And just deep, that space just deep to the arachnoid is called the subarachnoid space. So there's a space again filled with CSF here called the subarachnoid space. And then finally we have the pia mater. Okay, and we see the use of this term mater again. Pia means precious. Mater means mother. So this is precious mother. This is a very thin, delicate layer. We're going to come back to this, that literally covers the actual brain itself. So it's literally indistinguishable from the brain itself. Now, the idea behind the cranial meninges is that they're there to enclose and protect blood vessels that supply the brain. So they're gonna do that in addition to just protecting the brain. they're also going to contain and circulate that cerebrospinal fluid that we've been talking about. And in some cases, we're going to see that portions of these cranial meninges are going to form some of the veins that in turn help drain blood from the brain. There's a very unique system up there in the skull, and so the meninges actually play a part in now draining blood from the brain. So we'll show you that here in a few. Now the dura mater itself, we're going to break down these each one by one. The dura mater here is a double layer. It's got two layers. The superficial most layer is called the periosteal layer. Periosteal for the surrounding the bone layer, right? That's what peri means, peri for perimeter or surrounding, and osteal for bone. So this is that layer that's going to directly touch the bone. And there's a space between that, I'm sorry, there, just deep to that layer is the second layer of the meninges, and this one is called the dura mater, and this one's called the meningeal layer. Go mouse, sorry. So this meningeal layer here, right, we see is just one layer deep to the periosteal layer there. Right. Now, in some cases, you're going to see the periosteal layer and the meningeal layer split, and in between here you see this void or space, and inside you're going to see that that blue fluid that the cursor is, if we look at it here, it's currently pointing on. I moved it again, but here it is right here again. This space is called a dural venous sinuses. This is one of those places where the meninges come together and they actually help form those veins that help drain blood from the brain. Now, the space that we see directly beneath, and I'm going to move my mouse again, do we see this thin space here just deep to the meningeal layer of the dura mater? This is the subdural space. That space between the arachnoid mater and the dura mater. All right, so the arachnoid mater, you can just call it the arachnoid as well, again, lies immediately internal to the dura mater, right? So it's going to be just deep to the subdural space. And so we see that periosteal layer here and the meningeal layer here, and there's a dural venous sinus right in between. And now, here is that layer here, and here's that arachnoid mater. And notice that this layer often looks like a web, hence the term arachnoid. Now, the space just beneath this arachnoid mater here, the space where we see the actual webbing, is called the subarachnoid space, so just beneath the actual arachnoid mater. And these branches that make up the webbing uses one of these terms that we continually reuse, and that is the arachnoid trabeculae. This is a delicate web of collagen and elastic fibers that are found within the subarachnoid space. And in some cases, this arachnoid layer is actually going to pierce through or break through or erupt through the dura mater, the meningeal layer of the dura mater anyway, and we're going to get this arachnoid villus. The arachnoid villus is a projection that allows cerebrospinal fluid to pass within those venous sinuses, and this allows us to exchange some of the fluid of that CSF with the fluid inside the veins, i.e. the deoxygenated blood. All right, so that was the arachnoid mater. We can see the subarachnoid space, and then we've got this very delicate layer right here. And this deepest, most layer of meninges is called the pia mater. Pia mater, precious mother, this is the innermost layer. It's a very thin, delicate layer. It tightly adheres to every contour of the brain. So it's indistinguishable from the brain itself. Unlike the arachnoid, where when there are these depressions in the brain, the arachnoid will go over that depression rather than following into the valley of that sulcus, per se. I'm going to show you what those are in a bit here. So you can see that this layer, while very thin, it does actually just surround the entire brain rather than actually adhering and following every contour of the brain. But this is not the case with the pia mater. The pia mater will legitimately cover and follow every contour of the brain. Okay, now we talked about where the brain itself, the dura mater actually separates portions of the brain up. And this is a wonderful example here, right? So we see these flat partitions we call septa, where the dura mater itself actually extends now inferiorly from this top portion here and actually divides up the right hemisphere of the brain from the left hemisphere of the brain in portions of it anyway, right? That helps provide additional protection, support of the entire brain overall. Okay, so the ventricles of the brain. Ventricles are literally spaces. These are open spaces or cavities within the brain, which sounds weird. This is not filled with a tissue. It is literally an open space. And we see these ventricles here. They're ghosted out in a kind of a bluish gray color here. So we see this is one ventricle. This is another ventricle here to the side of it. This is a third ventricle here we're going to talk about. And then here's that fourth. So the four ventricles of the brain. They are continuous with one another. Think of this like a really odd cave system inside of a mountain, where you've got all these different caves connected to each other via tunnels, right? Tunnels here, or we see this other tunnel here, and we see this other tunnel extending down here into the spinal cord. So we're going to talk about all those. But you want to think of the fact that these ventricles are continuous with one another. There's four of them. There are two lateral ventricles. We see the right lateral ventricle here and the left lateral ventricle here. Now, there are two of them, and these are not numbered 1 and 2, so it's not like it's left first, then right. So this isn't 1 and this isn't 2. This isn't 1 and this isn't 2. There are two of these. They're called the lateral ventricle, and they're denoted by left or right. The third ventricle is literally called the third ventricle, and it does kind of look like a weird creepy duck in this picture. I'm not sure that it necessarily looks exactly like that in the way you're going to see it in the brain, but that's fine. This is the third ventricle. It is referred to as the third ventricle, and this is located within a region of the brain that we call the diencephalon, which you haven't learned yet, but you will. And then finally, connecting the third to the fourth, we're going to pass through this, what we call cerebral aqueduct. In this figure, it's also called the mesencephalic aqueduct. Same thing, different names. You'll learn it as the cerebral aqueduct. That's a lot easier to spell. Is the fourth ventricle here. And the cursor's on it right now, and as soon as my cursor catches up with me here, there it is right here. Now, the fourth ventricle lies directly between the cerebellum and the pons. And so these are two regions of the brain that we're going to discuss, and right between them is this fourth ventricle. Again, these are continuous. CSF is flowing, so cerebrospinal fluid is flowing through all of these ventricles. So it's flowing from one to the next very slowly, obviously, but nonetheless, there is exchange there. The cerebrospinal fluid is a clear, colorless liquid. It circulates within the ventricles and within the subarachnoid space itself. It's formed by a choroid plexus layer of tissue that lines each ventricle. And the choroid plexus is itself composed of these cells that we talked about before. These are a specific type of glial cell that we call the ependymal cells, right? And these actually produce, these ependymal cells actually produce this cerebrospinal fluid from the blood plasma. The job of the cerebrospinal fluid obviously is to protect the brain. It also bathes the exposed surfaces of the central nervous system. It completely surrounds it, and it provides some pretty important functions here. It floats the brain. So we talked about this before. Remember how I said the brain basically floats in the skull? Again, because of the cerebrospinal fluid. Brain actually floats in there. That provides some protection, right, from shaking around too much, again, within reason, against damage. But the other really big function that the cerebrospinal fluid serves is this idea of environmental stability. And that is that the actual environment that the brain is literally soaking in is important. Remember, the cerebrospinal fluid is made of things with ions and proteins and hormones and all these things like that. And so the idea is that CSF is maintained at a stable level. So that way the brain doesn't suffer from erratic changes in these concentration levels of certain things. Or heat. Here's a good example, heat. If it gets really hot outside, the nice thing about that cerebrospinal fluid is that it kind of buffers against that change in heat so that your brain doesn't get too terribly hot and overheated. pH, again, another thing. It allows for stability in the pH because, again, we expect the brain is going to need a stable environment in order to function. The blood-brain barrier, we talked about this before. This is that cutoff from general circulation. So the actual neurons themselves are cut off from general circulation. And so it allows us for a strict regulation of what can and cannot enter the brain, right? So it prevents exposures of a lot of things like drugs, waste being things like urea that we produce through normal metabolism, or waste products from food that we eat, or whatever the case may be, as well as, pardon me, as well as variations in levels of normal substances. So again, those ions, those hormones that could adversely affect brain function, we don't want that to happen. We don't want a fluctuation in an ion per se to really screw up the way our brain functions. Now, if you're a lipid soluble material, so we're talking about something that is hydrophobic, right? So it doesn't diffuse well in water because it's much more of a lipid-based molecule, then it can just easily pass across the cell membrane of the astrocyte, and so it can diffuse straight through the blood-brain barrier. But water-soluble molecules, on the other hand, things that are hydrophilic, things that dissolve very easily in water, these, on the other hand, must be what we call actively transported. This is the idea that we must use a pump, some kind of active pump, to pump this across from one side of the astrocyte to the other. And that puts the astrocytes in these positions to act as a gatekeeper. It holds the keys to the gates to allow certain things in and deny access to everything else. Now, the blood-brain barrier, by virtue of its function, by virtue of what it does, is noticeably reduced or missing entirely in 3 distinct regions of the brain. It's not seen in the choroid plexus. Remember, the choroid plexus makes CSF. So if we're going to be pulling substances from the like fluids from the blood to help make the CSF, we can't have that blood-brain barrier in the way, keeping us from doing this job well and efficiently. The hypothalamus, this is a master regulatory area for our endocrine system. We cannot have a blood-brain barrier in the way, keeping us from determining what hormones we have to secrete and what not to secrete. So noticeably reduced or missing in the hypothalamus. And then finally, the pineal gland. Pineal gland is really fun. It's one that makes for a really easy example to make. For those of you that either have kids or you have babysat kids or you have younger children in your family, or you know this well. For those of you that don't, it's actually a pretty funny thing when you become a parent, or at least when you get to spend time around younger kids. Little kids are amazing in the fact that they will just run and play, and they have so much energy, and then they start to get tired, and you can see they get crabby, they get tired. You can kind of tell they're getting tired, and then at some point, if they don't get a nap, then they get the second wave of energy that just carries them for a little bit longer after a while there, and they they function for just a little while, but at some point, and every parent can attest to this, it's amazing. Kids will just pass out. They're just going to pass out in basically whatever position they're in, wherever they're at, and it's impressive because sometimes they end up being passed out in these really weird positions, like half hanging off a couch, upside down in a chair. My daughter frequently sleeps in these what appears to be an incredibly uncomfortable position in her safety seat in the back seat of my car, and yet I couldn't sleep like that. But at some point, when they just get tired enough, that pineal gland in the brain, which regulates circadian rhythm, it regulates our sleep, our day-night cycle, our sleep cycle. This pineal gland just basically effectively shuts them off because they're so tired. Their body is saying, "You're done. You need to sleep." Here's a figure showing the astrocytes, right, which looks kind of like a weird octopus, I know, or kind of a weird sea creature like the kraken. And you can see how it actually attaches to these capillary networks and helps facilitate that blood-brain barrier. Okay, so we need to go through the regions of the brain, right? Because these are not necessarily as intuitive as we'd like them to be. Okay, so we've got the cerebrum here, and this is very easy to see. It looks very folded. Looks like it's got these hills, and then it's got these hills, by the way, are called gyri, singular being gyrus, and then these valleys in between. They look like creases, and they're called again, plural here would be sulci, right? Sulci. Singular would be a sulcus. That we see this valley in between here. So that's particularly easy to see. The diencephalon exists right here. Right, so we're going to come back to that. And then we've got the brain stem itself. And then finally, the cerebellum, which literally means little brain. So the cerebellum looks a lot like a mini version of the cerebrum itself. The brain itself has 12 pairs of cranial nerves that we'll discuss later that operate various different tasks. So again, we'll come back to that. Now, in an adult brain, this is important. An adult brain, because a child's brain isn't done forming yet, so it varies depending on the age of the child and whatnot. An adult brain has this extremely folded outer surface of the brain, and we see all 4 lobes here, well actually 5 lobes here that have all these gyri and sulci. Kids' brains don't look anywhere near like this because they're still developing. So it all depends entirely on how old the child is that we're talking about. We're going to start here with the cerebrum, going over these regions here. The cerebrum itself is actually divided into a left cerebral hemisphere, which the pointer is on right now, and then the right cerebral hemisphere. And they're separated into these hemispheres by this longitudinal fissure seen here in the middle of the brain. There are also transverse and lateral fissures that we also see as well. There's a transverse fissure that separates the cerebellum from the cerebrum, and there's lateral fissures that separate the temporal lobe on the left of the left or right side of the brain from the parietal lobe that we're going to see there. So again, I'm going to come back to, I'll point that out, but you have to have a lateral view of the cerebrum in order to see the lateral fissure there. Now, each hemisphere has 5 functional areas that we call lobes. Now, when I learned this, there was only 4, and so I'm going to show you the relatively new lobe that has been discovered recently, relatively recently anyway. So you've got the frontal lobe here on the right and here on the left. You've got the parietal lobes here and here. You've got the occipital lobe here and here. Now this one is occipital. This is a superior view of the brain, so it's actually much larger, and it does go farther down underneath the brain. So I'll show you that here, more of it in a second. We cannot see the temporal or the insula here, so I'm going to move forward for a moment so that I can show you. This is the temporal lobe here. And then this is a much better view here of the occipital lobe. Right? Don't pay attention to the coloring here. This is supposed to be for function, but uh, temporal lobe, occipital lobe, and since we're here, see the sulcus that exists right here, or outside this fissure. This is the transverse fissure that exists. The last lobe that you have not yet seen, this is a relatively new one, is this tiny little lobe here, just deep to both the frontal lobe and the temporal lobe here, and that is the insula right here. Now, the cerebrum is associated with all the very higher level functions of the brain. We're talking about conscious thought processes, so things that you're aware of consciously are going on up here. Intellectual functions, right? The idea of intelligence and whatnot, that stems from the actual cerebrum. Memory storage, memory retrieval, and complex motor patterns are all seen here. Now, there's a central sulcus that exists between what we call the motor and the sensory portions of the cortex. And so here, that cursor is basically right on that central sulcus. Come on, catch up with me here. There we go. I just need to get the cursor to catch up here. This is that central sulcus that exists right here. And we've got the sensory portion here that is just posterior to that central sulcus. And we've got the primary motor cortex here that is just anterior to that central sulcus. Now, there are motor and sensory association areas that we just got, motor and sensory areas we just got done talking about a little bit. We will come more into detail here about in a little bit. And we've got these association areas, right? So areas where we associate certain functions with. So we're going to come back and talk more about that here in a bit. Integrative centers, these are areas where a great deal of certain types of information is integrated so that decisions can be made based upon the data coming in, the stimuli coming in. We've got these basal nuclei that deal with subconscious control of skeletal muscle tone, or coordination of learned motor patterns. So, for example, subconscious control of skeletal muscle tone. The best way to think about this can be something like your basic posture. Everybody's got a basic level of posture, and that posture it's often something we don't really think about that much until, again, somebody brings it up to us or we start thinking about it, but most of the time we don't. We do still stand up straight. Those back muscles have to be being used. They're being told to do something even though we're not necessarily consciously aware of it. So there's a good example of that. Coordination of learned movement patterns. This can be things like how many of us have driven before, where, and this is one of my favorite examples, where you get in your car and you're destined to say go, you're supposed to go to, say, work, right? But for whatever reason, you're having one of those days where you get in your car, you start driving, and then you blank out. You're thinking, you're all stuck in your head, and you're all wrapped up in this conscious thoughts. And all of a sudden you kind of, and this is where quotes come in, you kind of wake up and you notice, why am I at Harper? Why did I pull into Harper? Because you've driven to some place that you did not need to go, but that because you were consciously, thinking about other things, you apparently just went to a default place. Now that Harper in this case can serve really any place that you find yourself ending up at. But obviously you were awake. I mean, you were conscious and awake and functioning to do all that driving. It's not like you got into an accident. It's not like anybody died in that whole process. You did a perfectly fine job of getting yourself to the wrong place that you were going. And all those coordinated learned movement patterns are the driving. You steering the wheel, making sure that you're giving just a subtle amount of gas and making sure you use the brake when it's appropriate and whatnot. Coordination of learned motor patterns. Now, here's the real fun one, and I really enjoy this because anytime we talk about emotion here, everything gets sticky and messy. Everything gets um, complicated with emotion. And the limbic system deals with the emotion, and that's found in the cerebrum again. The hippocampus, which is essential for storing, essential for learning, essential for storing long-term memory, is also found in the cerebrum as well. So this figure here is my favorite figure of all time because this figure it has been used in so many different textbooks since about the 1960s, and obviously it's been updated and changed a little bit, modified as we learn more and more about the brain. But this is hysterical because here on the left, highlighted in blue, this is the primary motor cortex, right? So this is this whole area here, this whole gyrus here that you see highlighted in blue up here in the upper figure. And this side here is the primary somatosensory cortex, right? So this is involved in sensation. And this, again, here's that central sulcus here. And this is that orange or rust-colored highlighted gyrus that you see here. Motor, somatosensory cortex. Now, the reason I like this, and I'm going to focus on primary somatosensory here for a moment, because this is the part that's kind of so often thought about. So back in the 1930s and 40s and 50s, when we had less, I shouldn't say we, but when there were less than desirable ethics and morals involved in, say, medicine, there were, and unfortunately, usually the patients that were used were often, in that time, mental patients, right? Patients that were in, at that time, were called asylums, right? Which obviously aren't called mental asylums anymore, but that's at that time what they were called now, instead of mental health facilities like they are today. And so these were patients that could not advocate for themselves, which family may or may not have ever been around at that point anymore, and whatnot. And so, in this case, uh, they would do brain surgery on an individual. So they'd expose a portion of the skull, and then they would take these little electrical probes, and they would gently or lightly touch certain portions, and the patient could actually tell them where they feel this stimulus. So, for example, if a physician were to place an electrical probe here, they would feel a stimulus or sensation on the forearm, right? That's how the patient would perceive it. Or if they were to touch here, that same stimulus would be on the nose, or here being somewhere in the lips, teeth, gums, or jaw, somewhere depending on where it was. But the reason I love this figure so much is if you look at the hysterical drawings associated here on the top here. So again, really huge hand, Mick Jagger lips apparently, super small stomach, and so on, right? But this is the map associated with certain areas on certain gyri associated with certain stimuli in certain places of the body. Now, conversely, on this side over here, this is the primary motor cortex. So if you've ever seen that Orbitz commercial where the physician is doing brain surgery and the patient has got a laptop on their lap, and you see the physician just touching places of the brain and the patient is typing in the search, this is what that commercial is alluding to. That if you place an electrical probe on some portion of one of these areas, like say for example here, the arm would move, or here the elbow would move, or here the forearm, some portion of the forearm would move, or here the wrist would move a certain way, or here the hand would move a certain way, right? So those electrical stimuli applied on this particular area of this gyrus would cause that certain area to move. Okay, the diencephalon here is this kind of middle region here. We see it associated between the epithalamus, which is the pineal gland that's right here, and the thalamus itself, which is this region right here. The hypothalamus, which is this region just below the thalamus, hence the term hypothalamus. And then finally, the mammillary bodies, right, which are not necessarily easily seen here. You can see it better on this figure down here. This is the mammillary bodies. So again, we're talking about this general region right here. Now, the epithalamus, this is otherwise termed the pineal gland. Either term is acceptable. I usually use pineal gland just because I think it's more common. And this regulates the amount of melatonin produced by the brain and thus released out into the body that helps regulate day and night cycles. The thalamus there, this is a major area of sensory information, for processing of sensory information, for processing of motor information, and for relaying of sensory information or motor information to the appropriate places that they need to go. The hypothalamus here, this is that it connects directly to the pituitary gland, which is right here. Pituitary gland is the master gland of the endocrine system, whereas the hypothalamus is the master control system. Right here, the hypothalamus is the master control area of the autonomic nervous system, and thus then the endocrine system as well, which this is often thought of as our homeostasis center, as well as a large source of our emotional behaviors. Finally, the mammillary bodies are not very big. These are pretty small. We see them here on just the inferior surface of the, inferior to, say, the thalamus, and just a little bit posterior to the hypothalamus. And the mammillary bodies are actually a small region of the brain that control feeding reflexes, especially early in life, right? So, the idea of baby suckling or swallowing. Swallowing is a reflex for adults, right? So those kind of feeding reflexes. The mesencephalon is otherwise referred to as the brainstem or the midbrain, so it's got 3 different names. Again, I'm more inclined to call it the brainstem because this is a whole lot less complicated to consider than the mesencephalon. There's a lot of things going on here. This is the basic portion of the brain. This is basic survival kind of stuff, as opposed to, say, higher level thought processing and emotion and all that other stuff. So within the brainstem, we find the cerebral nuclei, or sorry, not the cerebral nuclei, but the nuclei for processing visual and auditory information. So incoming visual stimuli, incoming auditory information, right? And these will in turn generate an involuntary somatic motor response to those. So let's say, for example, that you're joking around with a friend and they go to like poke you in the eye. They're not actually going to poke you in the eye, but it's kind of one of those joking things where they're going to act like they're going to poke you in the eye. And your knee-jerk reaction is to kind of flinch. Maybe you put your hands up over your eyes as well, or at the very least you close your eyes and kind of wince, right? That is an involuntary somatic motor response to visual information coming in. This is the idea that we're talking about here. Basic survival kind of stuff, basic protection kind of stuff. The brainstem also allows for the maintenance of what we consider to be consciousness. So the idea that somebody is in fact conscious is actually controlled in the brainstem. The corpora quadrigemina, in your lab manual, this is often called the tectal plate, is comprised of 2 pairs of structures, 2 superior colliculi and 2 inferior colliculi. Each individually is called a colliculus, right? So 2 superior colliculi, 2 inferior colliculi make up the corpora quadrigemina. And these are relay stations for visual and auditory reflex centers, right? So they're going to relay information, both visual and auditory, for the reflexes associated with the body. Whoa, too fast, too far. The red nucleus is shockingly red. And this is going to integrate information. This is going to integrate information from the cerebrum and cerebellum. And it's going to help in producing involuntary motor commands to help maintain posture. So again, we talked about this, the idea of maintenance of posture in some ways, right? So again, that red nucleus here is going to play a role in those actions. The substantia nigra, nigra means black, substantia means substantially. So substantially black, that is this black colored nucleus that we find in the brainstem. And this sends inhibitory information to cerebral nuclei to regulate motor output. And this is closely tied with the neurotransmitter dopamine. So the idea here that it helps us kind of fine tune our skeletal muscle motions so that we don't shake a great deal. And that is something that would be typical. We shake a lot more because of all this constant influx of information from motor output. But the inhibitory information that's sent from this area helps to kind of smooth our skeletal muscle movement so that they're much more fine-tuned, they're much more stable. Finally, the cerebral peduncles. These carry ascending information and sensory information to the thalamus. So when we've got, let's say you put your hand down on a hot surface, that pain that you feel, the original stimulus goes up from obviously your hand all the way through up your spinal cord, through the cerebral peduncles and into the thalamus, so that you can register this at a conscious level that, hey, ow, I just put my hand down on a hot pan. Here are the substantia nigra, and here is a red nucleus. So one substantia nigra, here's the other, and then here are the two red nuclei. Here are those colliculi for the corpora quadrigemina. Here are the two superior colliculi, and here are the two inferior colliculi, and all four of them together make up the corpora quadrigemina, or in your lab manual will be called tectal plate. Here's the thalamus, and we see the pineal gland here, but these are a part of the diencephalon. These are the cerebral peduncles. Okay, now the pons is this first bump. When you look at the actual brainstem, it's this first bump here. This houses nuclei for sensory and motor cranial nerves 5 through 8. It houses specific autonomic respiratory centers that have to do with controlling the rate and the depth of your breaths or breathing, right? So how fast you breathe or how deep those breaths are. This is going to be monitored and maintained in the pons. It's also going to modify activity of the respiratory center in the medulla oblongata. So actually respiration is actually controlled by both the pons that I've already showed you here and the medulla oblongata here. So it's actually a kind of a partnership between these two areas of the brainstem. And the pons actually connects to the cerebellum that is directly behind it. So we see this mini brain here, the cerebellum right here. The cerebellum has two hemispheres. The cerebellum, in a lot of ways, is a lot like the cerebrum. It looks a lot like it. It's got gyri, which are those hills, and the sulci, which are the valleys. It has two hemispheres, just like the cerebrum. So we see a hemisphere here and another hemisphere here, left and right. And this space in between is called the vermis. This is a connection point between the two hemispheres called the vermis. Now, function of the cerebellum, very important. This is the coordination and fine-tuning of skeletal muscle movements from learned patterns. These are learned muscle movements, right? The best analogy I can give to this is if you've ever watched somebody who can really dance and really dance well, if you've ever watched them dance, it's beautiful, it's fluid, it's graceful, it's amazing and impressive. And that is because they can move their body in such an intricately fine way because of the cerebellum. The cerebellum has become so good at learning this set of skeletal muscle patterns that it knows exactly how to move it just when, and they have such fine-tuned control, such subtle movements in there. So it makes it graceful and beautiful. It's also involved in equilibrium, so our ability to understand that we are in fact upside down or right side up, right, or balance, being able to have that maintain our balance, stay upright and walking upright. Again, the vermis is that connection point between the two hemispheres, And if we do a cross-section of the cerebellum, we see that it's got some gray matter around the external side, again, very much like the cerebrum. But inside, again, very much like the cerebrum, we've got white matter as well. Except here the white matter looks, for all better purposes, a lot like a tree. And so the term arborvitae is used to name this structure. This structure is called the arborvitae. Arborvitae literally means tree of life. And so hopefully that will help you remember it. The medulla oblongata is this portion here. It's that last bump there before we get into the actual spinal cord itself. This connects directly to the spinal cord. It's a relay for sensory information to the thalamus, which if you're kind of following up here, the thalamus would go all the way up right about here. We've got a lot of autonomic centers controlled here in the medulla oblongata. We've got cardiac for both rate and strength of actual contractions. That is controlled here in the medulla oblongata. The vasomotor, so we're talking about vessel diameter control in response to blood pressure. That is controlled here. Respiration, again, controls rate in cooperation with the pons. And then other very unfortunately uncomfortable kinds of things that we have to be able to do: coughing, sneezing, salivating, swallowing, gagging, vomiting. These are all actions that are controlled here right in the medulla oblongata. It also houses the nuclei for cranial nerves 8 through 12.